Next speaker is going to be Dr. Karyn Havas from Pipestone Veterinary Services. Dr. Havas is going to present to us a summary of the most recent results they have had with new alternatives for sampling of pigs. So, thank you, Karyn.
Thank you. Okay. Well, thanks for having me today. My name is Karyn Havas. I'm the Director of Veterinary Epidemiology for Pipestone Research. If you're not familiar with Pipestone, Pipestone is a large swine production company stationed or located here in Minnesota with customers in South America, Latin America, and Asia as well. So we do a lot of different things in trying a lot of different things. We like to innovate. We like to trial different methods and different products. So today I'm going to talk to you a little bit about two programs that we ran. One is a research project on a novel sample type, and another one is just trialing on-farm surveillance. So we don't have too much time, so let's dive in. So specifically, the first topic is about this meat juice study. So I had a project in Uganda. I was working with Makerere University, and we got some funding to layer on evaluating a meat juice study. Because meat juice, using the diaphragm, is a very low-cost sample type. It doesn't have a lot of value. If you have to do a lot of sampling at a slaughterhouse, it's a possible sample type. Well, even easier potentially than meat juice was muscle swabs of the same tissue. So we evaluated meat juice and muscle swabs against spleen and spleen swab samples. And the second topic I'm going to talk to you about is the swine testing for ASF/CSF monitoring program called STAMP that we ran right here in Minnesota in the Midwest for on-farm mortality on a variety of farms, grow, finish, and sow farms. So let's start with let's start with meat juice. So the purpose of the study was to do this comparison because in the United States, spleen and spleen swab samples are gold standard and allowable to be used. And so we wanted to compare to the USDA gold standard, okay? I did it with this team over here. This is the team from Makerere that I worked with, and it is a published paper. Oops, excuse me. It is a published paper. You can get it through Journal of Veterinary Diagnostic Investigation. If you can't get it, just email me and I'll send it to you. So we knew from previous work or for our other ongoing work that there was a lot of ASF in Uganda slaughterhouse pigs. I mean, a lot. I think our other study had like 59% of the pigs were positive. So we knew that if we targeted pigs with some clinical signs, that we would probably have a lot of ASF positive pigs. So any pigs that had at least two of these signs, diarrhea, ecchymoses, enlarged or hemorrhagic lymph nodes or spleens, or petechiations were selected for the study. We ended up sampling 500 pigs, and we needed 139 to be able to identify a 90% sensitivity of the sample type. We used the ZASC assay, which is a real-time PCR assay that's used by the Foreign Animal. Yep, it's working. Okay. Foreign Animal Disease Diagnostic Lab. And it's they're a OIE reference center for ASF. It's a well-validated, pretty darn sensitive assay. We also looked at pools because everybody wants to pool because it saves resources and time and supplies. So we looked at 72 pools, and that allowed us to evaluate those sample types with 95% sensitivity, with 95% confidence, 5% error. And we decided to do this for the most ideal pools. We wanted to compare the sample types with their most ideal. So we used this lowest CTs, the samples with the lowest CTs from each sample type. We took 1 positive and 4 negative and put them together. So what did we get? We were actually really surprised. Meat juice had the highest positivity rate far and away. We don't know why, to be honest. But it was we had a 72.4% positivity compared to spleen swabs, and muscle swabs, which had 50.1, 48.9, and 44.2. And it was statistically significant. The confidence intervals didn't overlap between meat juice and all the other sample types, which did overlap. The spleen swab, or the spleen did have the lowest CT values, meaning that it had probably the highest viral load, but the meat juice had the highest percent positivity. And that was really interesting and no one expected. Genotype 9 is circulating in Uganda, and so that might be one of the reasons for the differences, but we're unsure. As for the pools, spleen did perfectly, because that's what spleen does, right? Like spleen works really, really well for ASF, and all the pools came up positive. Meat juice didn't do bad. Only one of the pools was misdiagnosed as a negative, and muscle swabs did a little worse, with 5 of the pools being misdiagnosed as negative. And clearly, because we chose the lowest CT values for each sample type, spleen had the lowest CT, the pools had the lowest CT, and then meat juice, and then muscle swabs. So this gets into the really interesting part is, well, why did meat juice have the highest positivity rate? Like significantly higher, 22.4% higher than the other ones. And we kind of went through the process in our discussion of trying to figure out why that may be. It could be genotype. A lot of work is done in genotype 1 and genotype 2 because those were the 2 genotypes that circulated outside of Africa. There's not always a lot of work done on other genotypes. So genotype 9, like I said, is typical in Uganda. They have had a genotype 10, but it's been a very long time since they had a sample collected that's genotype 10. There's not a ton of sequencing, but we did sequence 31 isolates this past year from different regions, and it's still genotype 9. And that's probably the largest, most robust sequencing event in a long time. So that might be why. That potentially could be a reason, and it highlights that maybe we do need to look in between genotypes. And then we went through, well, these would be false positives if they're inaccurate. And there's a couple reasons why you could have a false positive. Did we contaminate the samples in the field? Right, first question. Well, every sample had a new pair of gloves used, a new piece of equipment used. So if we had to use pick-em-ups and a scalpel blade, each sample got their own set. New swabs, new everything. So, and we would have seen contamination across the board, right? We would have seen a lot more contamination with all sample types. So that doesn't seem likely. Was it contamination in the laboratory? Well, all of our negative controls, our amplification, our extraction, we used a positive and negative amplification, positive and negative extraction, and a xeno internal control. And all of the controls worked beautifully. And again, we didn't see issues, any odd results between sample types. So there wasn't anything that made us think that there was any lab contamination. And then you go, okay, well, maybe there was something with the sample itself that made it result in a false positive. And it doesn't really make sense because we know like heme and myoglobin are inhibitors of PCR typically. It's why blood's actually sample to use with ASF diagnostics of all of our approved sample types. So that doesn't really make sense that you'd end up with false positives. You would expect false negatives. So we don't know why meat juice was the best. It very well might be an ideal sample type, and I think more work needs to be done. I think it's very exciting. I think it's very hopeful, and we'd like to see more work done. We attempted to do a little more work. In fact, Dr. Shambo and I were working with a few folks, a few other colleagues to do more work on genotype 2, and it kind of fell apart because of vaccines. But genotype 2 really needs to be looked at in field conditions. We also had challenges that might have influenced this, in that Uganda does not have animal welfare requirements, right? There's not a shipping duration time. There's not a certain amount of time that within that you have to provide animals food and water, things like that. So many of these animals, not many, but a handful of these animals ended up being severely dehydrated. So when you took the meat juice, when you took the diaphragm and you freeze-thawed it to get the meat juice sample, you didn't get anything. So what we decided to do is add a little phosphate buffered saline to those samples, massage it into the muscle, freeze-thawed it again, and it actually worked really well, and see if that would allow us to do the test, and that worked really well, actually. But those didn't account for all of the additional positive tests or positive results. Oh, one other comment on meat juice is we're always talking about lab capacity, right, and workflow, and the comment is swabs are the best, right? Blood and swabs. Either you take a pipette and you draw out your sample, or you take out your swab and you draw out your sample. There's no other processing. Those are the best, far and away. I don't think meat juice is all that hard. It would create a change in workflow, but it's just a freeze-thaw cycle. So you would literally have to freeze a sample and then thaw it and take your aliquot. Whereas tissue processing, if you ever process spleen or liver or kidney or any of the tissues, it's a process. It's a full process. So this is an easier sample type than, say, a spleen sample or a lymph node or a tonsil. Okay, that's the sum of my first project. We're going quite quickly because there's a lot to cover. But of course, these are the funders. The National Pork Board provided funding for this project, and then we benefited from another project funded by the Department of Defense that provided equipment and training on these protocols that we used. And of course, I'd like to acknowledge my colleagues at Makerere, Bonto Fabre at FADL, and John Ekakoro, who was working with me as a postdoc and is now at Kansas State University. Okay. So we go from diagnostics, trying to find new diagnostics, because the more the better in some ways, to how do we do on-farm surveillance testing? Does it work? Is it feasible? We can't send vets everywhere. Can on-farm staff do it? And we were having this discussion with Lisa Rochette and John Korsland and the Swine Commodity Group at USDA, along with South Dakota State University. And we ended up getting a project going where we looked at on-farm mortality, where farm staff picked up the samples. For about 2 years, we did this project. So it came in 2 phases. The first phase was, we had 8 farms with 25 sites enrolled, 3 of which were sow farms. They were all trained on how to do this low exposure spleen sampling. So one of the problems about mortality testing is no one wants to open up a carcass and contaminate your whole site. So we developed a method, actually Brent Pepin came up with this, Dr. Brent Pepin, doing a low exposure spleen sampling where you make a small incision on the side of the animal and pull out the spleen. And I'll show you a picture. The second phase occurred in August 2022 to February 2023 from our most reliable participants, right? So 3 farm enterprises with 14 sites between them, and we added in oral fluids so that we could evaluate, the lab could evaluate what it was like to manage oral fluids for African swine fever and classical swine fever testing. We use a case definition provided by the USDA Integrated Surveillance Plan for ASF and CSF testing. So any pig that was submitted as part of this program that died on farm had to have one of these clinical signs or sets of clinical signs. And they sent in their first 5 pigs every month that met that case definition. So these aren't the best pictures. I apologize. They're from videos that we made for the farms. But basically, low exposure swine sampling is the right side of the animal's on the ground, the left side is up. Halfway between the back and the belly, you make a small incision along the rib cage, you reach in, you grab the spleen. And it was actually really easy to do. People did it fairly well. And then you don't open the whole animal. You have some contamination, but it's not nearly as widespread as if you open the whole carcass. We did provide incentives. The first part of the program, they got $80 if they sent in 5 samples every month, and the second part of the program, they got $120. They got basically a credit towards diagnostics to try to get, farmers need a reason to be involved, right? They're busy people. You have to give them a reason why it's of interest to them. So I think one of the most interesting things about this were, why did people submit samples, right? Like what case definitions, part of the case definition did they meet? And lameness far and away won. And no one's surprised at that, right? So lameness could be a neurological issue, so it meets the case definition. And then off-feed and lethargy, which I actually think is really great, because if you talk to our colleagues in China, when they're testing for ASF on a daily basis, looking for ASF early, it's pigs that are off-feed and depressed. So that's essentially what we did here too, which is really nice. But then you have also cases that are of acute disease. You have paddling. You can't read this one, it's down here, but it's increased mortality, fever, off-feed, and widespread skin redness. Really glad we captured that pig in this program, right? That's a good thing. And then of course, widespread skin redness, increased mortality. One farm that was trained did not submit any samples, even with the incentives. One farm that was trained sporadically submitted samples. That might be generous. And the rest of them did pretty well. So overall, it wasn't 100%, but the farms that were enrolled and trained and involved did participate. We asked them a few questions about this, like how hard was it? So for low exposure spleen sampling, and the very small number of responses, right? So this was an anonymous voluntary survey. There are 6 responses. So there's still a lot of work to do, but basically the nursery pigs were the easiest to collect the low exposure spleen sampling on. They're very small. It's very easy to reach your hand in the abdomen, pull out the spleen. Whereas a sow, 400-pound sow, there's a lot to search around for. So overall, there were about 2 respondents from the sow and gilt, and I think 5 respondents from nursery here, or 6 respondents out of the nursery, and almost all the nursery guys didn't have much of a problem. Most of the sow guys didn't have much of a problem, and the market barns were a little bit split. They had a little bit harder time with it. And then when you ask them based on ease, manpower, and time, which sample would you like to use, and they could choose a few, they chose, the groups chose spleen as a majority. So in total, 4 people said the spleen was really easy to sample, and then 3 said they'd prefer oral fluids. So really, it might be as at least equivalent in terms of ease, manpower, and time as collecting oral fluids. Hard to collect oral fluids off of a dead animal, though, right? So, okay, so part 2. Part 2, we added in oral fluids. Everything else was the same. The thing that changed with oral fluids is you can't collect oral fluids from a dead animal. Really hard to get them to chew. So what you did is you took oral fluids from the pen where the animal died, or if they're in a crate, from their neighbor, right? So the crate next to them on either side. Just choose one. And we ended up with 295 oral fluid samples and 300 spleen samples because some pigs came from the same pen. Some pigs that died came from the same pen. This was a consistent group. We knew they would be consistent. They sent samples consistently over the course of the entire project. And again, we asked a few questions. And it's a smaller group, and we got a smaller number of responses. But basically, what we learned was that for nursery pigs, forget oral fluids, right? And I think we knew this. Nursery pigs are not easy to get oral fluids from unless you do a lot of training. Spleen was much easier. Oral fluids were preferable for the market pigs. But overall, a couple of the producers responded that both were easy, but they thought that the industry would prefer oral fluids for market pigs. We also enrolled this group in the Certified Swine Sampling Program as part of the incentives. So we offered all of the training, and all of them went through the didactic training, and 2 of the enterprises went through the entire training. So the enterprises that went through the entire training and got completely trained, they said it was a very good program. They learned things they didn't know, that they hadn't expected, and that with the knowledge they have now, if it hadn't been offered to them, they would have requested the training. So I think that speaks highly of the certified training, but I think we also learned from the one group that wasn't able to complete it, that around harvest, a PRRS outbreak, and other demands on producers, it can be really hard to get that done even when you're constantly reaching out and saying, hey, do you want to finish this? So I think there are challenges in just terms of time management for that. Basically, simplicity matters for on-farm mortality sampling. The forms have to be simple and mostly filled out for the producers. They need a checklist to make it really simple. This takes time. It needs to be worthwhile. This is manpower and time and resources. And the Certified Swine Sampler training is an excellent training. They found it really valuable. But again, I think veterinarians have a hard time finding the cost benefit at this stage. It's a preparatory thing, so preparation is often underfunded. And so that's a real challenge now, is to make that a value for farmers and veterinarians to get that done. This was funded through a USDA cooperative agreement that was administered through South Dakota State University. And there was a large team that worked on this, so very grateful to South Dakota State, very grateful to the USDA. I should have Lisa Richetts and John Korsland and Nikki Humphrey's name up here. I apologize for that omission. And I think that's it.
We have time for 1 or 2 questions for Dr. Havas.
Have you looked at testing tongue tips for ASF? They're much easier to collect.
The tongue tips?
Yes.
I have not. I know that there's a call for proposals out right now to do tongue tips. They are easier to collect, you're right, but I have not done that work.
How did you collect the meat juice? I'm sorry.
Oh, yeah, I didn't really go through that. So we basically took a 4 centimeter by 4 centimeter square of the meatiest part of the diaphragm, which took a little training, and then you freeze it and thaw it, which takes maybe 30 minutes at most. If you have a negative 80, it takes very little time. And then the exudate, when you thaw it, that breaks down the cells in the muscle, release juice, and that's meat juice.
Thank you.
I'll try to be quick, Karyn. Do you think the stage of infection in each animal when you collected the meat juice has a role to play in the level of detection?
Always. Yeah, these were acutely ill, right? They had clinical signs, active clinical signs. So absolutely. We didn't see a lot of chronic or inapparent infections in Uganda, to be honest. There's no evidence from our study that there's any chronic disease. So this was about acutely ill animals. Great question. Yeah. It was a 1 to 5 pool, 1 positive with the lowest CT value available and 4 negative. Otherwise following FATL protocols for pooling.
On the second set that you talked about, is the end game really to kind of just make it, you know, if African swine fever virus is here, to have options, I guess? Is that the game plan? Like, you know, you'd have, let's say, these two, but then also maybe tongue tips, or these are things that you guys are testing to try and make it as easy as possible for people.
Are you talking about the meat juice sample, the end game?
Any of them, I guess, right now. You know, what you were talking about,
Yeah.
kind of, is that the end game to say, you know, whether it's oral fluids or whether you're pulling the spleen out, or what that might be, kind of how easy is it to train on-farm staff to do this, right? Because there's not going to be enough people to do it otherwise, right?
Well, the end game for us was we recognize that trade is developing and changing. So regionalization is becoming important. Compartmentalization is a huge discussion, not yet affordable, but huge discussion. And we believe some of these things will have to be driven at the packing plant level. So if I'm a packing plant, what's the easiest thing for me to collect? So that is what drove the meat juice, but is also what drives the tongue tip studies or the ear tip studies. And so we were trying to contribute that way. And then we also feel that if we end up having ASF in this country, there's probably going to be some level of mortality testing for if we regionalize. We feel that way. There's nothing from USDA saying that, but that's a pipestone just if we had to predict the future. And we wanted to understand what it would look like to have to conduct mortality testing similar to MPIP, right? Dead, sick, and then healthy animals are tested in that order. So how would that look like on a sow farm or on a swine farm?
So it's really like, what's the easiest way, or, you know, start doing the testing, I guess, to be able to establish some alternatives to maybe what we have now for dead animals versus live animals, you know?
Yeah, would there be different options? And STAMP allowed us to just trial it and see how it would work. Like how it could actually be implemented with farm staff, because vets are not going to go out that regularly and collect samples. There's just not enough of them.
Right.
So.
Does it have to be, sorry, just one really quick question, I apologize. Let's say that if we did have African swine fever here, could USDA, since some research has been done on some of these types of samples, right? Can USDA enact like an emergency type thing or something like that, so you could, I mean, that may not be approved by them.
I wish Christula was here. So they would still have to do their own validation for trade. And it's all driven by trade. And I don't criticize them for this. I'm glad they do the work, because trade partners need to recognize our testing programs. And that's based on OIE validation methods. So I think they have plans in place to rapidly evaluate things should we be in that position. But I don't want to speak at all for USDA.